And smile.
That was great.
My boss was there.
I was smiling because of the line.
I should have a glass. Finish it in 45 minutes?
I'll try.
Anyway, I'm truly delighted to have John Lederer with us today to open our seminar series for the fall. And of course, John doesn't need an introduction to this audience. But we met the first time, I don't know if you remember, but it was many, many years ago in an NIH study session reviewing grants. At the time, the funding was good. It was a pleasure to be on the study session. John received his MD and PhD both from Yale, and did his PhD with Dick Chen. He then did clinical training at the University of Washington in Seattle, and spent 2 years after that, 2 or 3 years with Dennis Noble, when it was really exciting times in Dennis's lab. People like Dario Di Francesco was there, and I remember all the memories that we've seen in Montpellier when we had this meeting, which was great. John is very well known for his work in calcium cycling and calcium signaling in cells in general, including cardiac cells. And in fact, he's been a pioneer in looking at the quantum nature of calcium release from the sarcoplasmic reticulum in terms of sparks. And I was very happy to hear that calcium release from the SR is quantum, because my background is in quantum physics, so that reassured me that everything has little packets of release or uptake. Anyway, from sparks, John continued to blinks, and I don't know what the future will bring, but he'll tell you more shortly. Thanks for coming.
First of all, I'd like to thank you for the invitation and this opportunity to talk and drink with friends. If anyone needs, there's more wine here. I'll not drink it all, probably. I thought I'd start with this introductory slide. This is a painting by Emil Nolde, who was a German-Danish painter, who was a Nazi sympathizer and supporter of masks. And the reason I'm showing it is that things aren't what they seem. Masks reflect that, and Nolde was also, he was denounced by the Nazis and was one of those painters who was put on the almost untouchable list of painters because of his work on abstract art, despite his support for the party. So things aren't what they seem. And calcium sparks aren't always exactly what they seem either. There are parts of the talk that I simply had to cut out because of time constraints, and the number one issue was something that I would have kept in had I heard the introductory comments before. We have a lot of evidence today that the quantum nature of sparks is not quite always so quantum, but I'll have to come back and talk about that. It's a very interesting aspect of the story. There's subsparks and other components of sparks, I guess, like quarks. Okay. The work has been done with a lot of support from others, and that is bright. And I just wanted to mention Eric Sobey, Stefan Lennart, with whom we've done some modeling that I'm going to talk about. I'll present the qualitative version of the modeling. We're doing the more quantitative work now. And I forewarn you, even for those, many of whom do modeling as they live and breathe here, it may be possible that the sees in the presentation will be a little confusing, so I'll try to be careful. It's just a space that we don't normally live in. Andy Marks initiated some of the work because of all the controversy that he's generated, and we have some comments. Rocky Cass, Brian Hagan, Letty Gomez, Civic Guadalupe-Seaman, Julio Altamirano. And so it's a real pleasure for me to be here to talk about the work. I'll try to go pretty quickly because if I follow my normal tendency to talk excessively, it'll last too long. But I would encourage you, despite that warning, to interrupt me with questions and comments as I go through. It's more exciting for me and it may be more interesting for you. So if I say something that you disagree with, and I think I will say a few things along those lines, stop me. I wanted to also point out that I'm presenting some work by Didier Brochet and Peace Cheng on some recent work, and some of the sub-spark data that I can't show was that I took out last night was from them, but the blink work was all done in conjunction with those fellows, and the funding is to be acknowledged in tough times. If anyone's interested in this work, postdocs are available. Pretty exciting work. Baltimore's still a very nice city, a more livable city than you may think. And working on heart failure, arrhythmia, stem cells, sparks, blinks, etc. Mathematical modeling, and with diverse funding. Anyway, today I'm going to talk about a problem, the organization of the cell, calcium sparks and calcium release, pump leak balance, which even for modelers, many of whom are here, people who think about calcium flux quantitatively, some of the simplest notions of pump leak balance are occasionally ignored, at least based on presentations that I've heard. I'm not accusing anybody here. Calcium signaling complexity, and based on some of the conversations I've had this morning, I'm going to show some older work, but you'll see that the complexity of calcium signaling is remarkable in subspaces and in a time-dependent way. Talk about calcium blinks, and then the modeling data that hopes to resolve the problem. And the issue centers around this distinction, which I've been pushing lately, and I hope by the end of the talk you will agree it's a useful distinction. The distinction between the open probability of the ryanodine receptor, PO, versus P star, which we're trying to put forward as an intrinsic opening probability of the ryanodine receptor that is under normalized conditions. Just to give you a sense of what I mean by that, if a ryanodine receptor were to be phosphorylated or mutated, its intrinsic opening probability could change when other conditions were constant, as our calcium content or cytosolic content. So that's the idea of P star. So we want to have a tool like that in our modeling array so that we can refer to differences in ryanodine receptor behavior. And then I hope to present some sort of resolution of the problem. So the T-tubules are plentiful. They lie along the Z line. You can see them in this picture taken from Costin's work. You can see them on this scanning EM. The sarcomere is about 2 microns wide. T-tubules are plentiful, about 1 micron separating one from the other. And they form this interrelated intercellular array. If you fill the extracellular space with di-8 anabs, a little dye, you can see the T-tubules in a plane focus. They show the sarcolemmal membrane. The nucleus is excluded. And if you zoom in on a region, you can see the surface membrane. You can see T-tubules in the plane of focus. You can see T-tubules perpendicular to the plane of focus because they form a bright circle in and out. And you can also see the longitudinal components of the T-tubule system when they're in the plane of focus. And all of these things are corresponding to that, ryanodine receptor clusters are formed, as I saw on someone's blackboard this morning, 10,000 roughly per cell. You can see them in one plane of focus. In fact, you can count them up and you get a number close to 10,000 per cell as well. And the important point here is that the clusters are about 100 Ryanodine receptors on average in size, and they're formed right against the T-tubule membrane, and they are sandwiched on one side by a mitochondria that spans the Z-line to Z-line, the sarcomere, and on the other hand, the T-tubule. So the Ryanodine receptors are plentiful, and they form a paracrystalline array at the junctional SR. To show you this, let's see if this is plain today. This is from the group in Auckland, Peter Hunter's group. That this is a simulated part. It's not playing out well. Each heart cell is 150 microns long. You can see the Z lines as you saw before. The T-tubules are normally about 300 nanometers in diameter, and you can see some of the important details. For example, we were talking right before I started drinking the wine about the mitochondria that span from one Z line. You can see the Z line here, and through the T-tubule to the other Z line. It's about 2 microns long. And on the other side, you can see the junctional SR, and this is a typical cross section. The junctional SR. It's about 25 nanometers thick, forms a pancake that's 100 to about 300 nanometers in diameter. It wraps around the T-tubule, so this thin pancake is wrapped around, and in between you can see these dark structures, which are the Ryanodine receptors, and we believe the L-type calcium channels are placed on the T-tubule membrane so that they can fill subspace with calcium when they open. So that's our so-called subspace, the space between. If you zoom in, you can see there are roughly 8. So in this cross-section, you would, at a minimum, guess maybe 64 Ryanodine receptors were in this cluster, if this is just a random cross-section, something like that. And with the L-type calcium channel there, when it opens, even briefly, for a tenth of a millisecond or half a millisecond, there's enough calcium to diffuse throughout the subspace. And when it does that, a lot of the time it will trigger one Ryanodine receptor. When that one Ryanodine receptor opens, calcium will continue to fill the subspace and trigger the others to produce a calcium spark. You can measure the SR calcium content. Quantitation is a bit tough, but you can measure it using MagFlow4. MagFlow4 is a low affinity indicator. It's a little difficult to do it in the small rodents. It works really well in rabbits. And you can see that the structure of the SR and the ER is well represented. You can see that the ER is full. We get the impression that in the gold sheet, perhaps, there are also concentrations of calcium. This is a little, we haven't actually cross-checked this with double staining for Golgi, but it has the same structural appearance. And the details can be seen if you look for them, and you can see clear Z-line structures of the junctional SR and other components, longitudinal components. So the calcium sparks, we think, are the elementary release events. So when the release occurs, they're emptying the SR. That's going to be the blink. I'll show you that in a few minutes. They are synchronized by the electrical signal to produce the elevation of calcium, and you can see this sort of event that occurs. The sparks occur, these are rodents, so there are a lot of sparks in rabbits and in humans and in dogs and cats. There are many fewer sparks that occur during the diastolic period that you see here. So frequently in the steady state and diastole, in these larger animals, you see no resting sparks. Whereas in rats and mice, you see plenty. So this is a rat.
This is an unlimited cell, so it just springs back to its length all by itself.
That's correct. It has an internal spring, if you like, which is the structure inside. If you're interested in some of the biophysical details, when Colin was in the lab, Ernst Niggli made these measurements of how you can cause a Riger contraction that just starts, and if you flash ATP to produce a sudden jump, then you get re-extension, and you can measure the biophysical properties under those conditions. So yeah, it's an internal spring, as long as you don't shorten too much. So with respect to calcium-dependent arrhythmias, there's a problem, and that's sort of the content that I'm going to focus on now. How do you get calcium-dependent arrhythmias? And the problem comes about because those things that tend to produce calcium-dependent arrhythmias are self-correcting. And when they're self-correcting, they take away the driving force. Interesting. They take away the driving force for the process. So for example, if you have arrhythmias that are attributed to calcium waves in myocytes, you can see lots of data to support this. So you get high calcium, calcium-induced calcium release propagating, producing a calcium wave. This is the cellular equivalent of an arrhythmia. Arrhythmias are seen during SR calcium overload, so there's high SR calcium. So that's what we always thought was a requirement. When ryanodine receptors become more sensitive to cytosolic calcium, that's when we think that SR calcium goes up or some other feature occurs. Then we end up getting more spark behavior, and this is supposedly one of the underpinnings of calcium-dependent arrhythmogenesis, again, consistent with the above observations. But the problem is that in pathologic circumstances, when calcium-dependent arrhythmias are actually seen, the SR calcium is low. So SR calcium is low means that the sensitivity of the ryanodine receptors due to the high SR calcium is not there. So there may be something else, but that part, is, if you like, is self-correcting. And the idea is if there is a tendency to produce more sparks or more release, then it's going to drain the SR of calcium content. And that is the steady state of the circumstance that favors calcium-dependent arrhythmias. So the contradiction is that we know that high calcium in the SR produces arrhythmias, but in the cases where there are calcium-dependent arrhythmias, the SR calcium is low, so we don't have that driving force. And in fact, when we look at lots of different conditions like heart failure, myocardial infarction, genetic mutations, particularly in the ryanodine receptor, calsequester, and so on, in these cases, SR calcium is low in the steady state. So one of the issues has to do with this dilemma, and this may or may not be an issue for you when you think about it if you presented this, but it's an issue in the literature, and there's been a fierce debate, maybe too strong, but it's pretty animated about what causes calcium-dependent arrhythmias. So the paradox to resolve is how can calcium-dependent arrhythmias occur when calcium is low? And the one thing we know that is different is that the open probability of the ryanodine receptor is increased under these conditions. Now, this is why I have to invoke P star. What do we actually mean by that? Well, we started off, and many people have written on this, saying that that ryanodine receptors are more leaky, or that they're more sensitive to intracellular calcium, that is cytosolic calcium. However, this very leakiness is what produces the low steady state SR calcium, so it's self-correcting. So that's the problem. And David Eisner has been a real champion of this position that it's a self-correcting process, therefore it doesn't contribute to arrhythmogenesis. And and there have been editorials and comments on it, work in the literature. So I thought I'd first of all show you an example of calcium-dependent arrhythmogenesis with sparks leading to waves taken with a 5 Live. This is just a calcium overloading cell. But you can see the sparks are more plentiful. They're occurring spontaneously at a high rate. Spontaneously meaning that they're not triggered by depolarization. And then there's a gradual rise of calcium. So in this case, we're not too surprised. The high calcium that occurs seems reasonable. There's a high external calcium. So let's talk a little bit about pump leak balance, and I'll do this pretty quickly. So the problem in pump leak balance arises, so our problem that we're talking about, this paradox, because the probability of the ryanodine receptor goes up when it's phosphorylated or there's a mutation in the ryanodine receptor itself. It's self-correcting because as this probability goes up, there's a larger leak, and this loss of calcium reduces the content. The reduction reduces the open probability until it was before. So the leak must equal circa pump influx, always. So the pump has to be in balance with the leak. So if you have higher leak and you have a fixed pump, then that's why the steady state is what it was before. Okay? So the only thing that really has happened is that there's been a change, an apparent change in the sensitivity, the intrinsic sensitivity of the ryanodine receptor to calcium. So the pump leak balance, so fairly straightforward. You have in the cytosol, you have calcium entering the cell from the extracellular space, primarily through the L-type calcium channel, or the dihydropyridine receptor. So that's the leak. And the pump, in this case, is mainly NCX. Now, when I say this is the pump, there are other pumps too. I'm not ignoring the surface calcium ATPase, but these are the main pumps that leaks. So in the steady state, intracellular calcium depends on that balance. So for example, a rapid heart rate would produce an increase in the resting calcium because, if you like, the leak is up. And that's the solution to that pump leak balance. There is a change, the cost is a higher resting calcium. For the sarcoplasmic reticulum, you have a pump leak balance as well, where the pump is circa, pumping it in, and the leak is the ryanodine receptor leaking it out. Of course, there are other pumps and leaks possible there too. The other leak could be the IP3 receptor or other channels. And one of the possibilities, of course, is circuit will run backwards if that's in your model. So calcium exits the SR mainly by the ryanodine receptors. It's pumped by circa. And in the steady state, the exact amount depends on the pump leak balance. So increased calcium in the cytosol would increase circa more than increases the open probability of the ryanodine receptor, and so it leads to an increase of SR calcium content. If you block ryanodine receptors with tetracaine, so you block the leak, you increase content. So I guess everything seems obvious in terms of pump leak balance. As obvious and simple as it is, it's frequently overlooked in discussions of these issues. What we're going to focus on isn't just pump leak balance, but it's this question of transient versus steady state. And again, something remarkably simple, but overlooked by some of our esteemed colleagues. What are the time differences? So calcium is regulated in the steady state and also dynamically. Steady state regulation can mean anything you want it to mean, but in this case it means regulation in the order of seconds to minutes, whereas the dynamic regulation that we're talking about for the calcium sparks and calcium leak is milliseconds to seconds. So it depends on your definitions, of course. So what about the complexity the regulation of the ranitidine receptor open probability. Now, there's controversy. That doesn't necessarily mean there's complexity, but there is intrinsic complexity, and we'll talk about some of the complexity issues, and I think it will have bearing on the models that I've heard talked about today, but also has some bearing on how you think about calcium leak. So the dynamics of calcium do affect the open probability. The phosphorylation of the ranitidine receptor affects open probability. And both affect what we will consider normal function, leak, and arrhythmogenesis. Even though these are really overstudied topics, when it comes down to the modeling component and how you try to quantify each of these issues, the dynamics are a little surprising, even though some of the work has been published some time ago. So I thought I'd show you this one example because this is one of the few examples in which I have to say every time I show it, even to some of my colleagues who I've shown it to a few times, they are surprised. So let's try. This is a bilayer experiment in which we're going to pulse light to produce a photorelease of calcium in a region on the cis side of the bilayer, and on the opposite side, the luminal side, the trans side, we have conditions that make this like the SR. The ranitidine receptor is placed in the bilayer. Cis side is like cytosol in terms of ATP. It could be higher or lower. Magnesium, free magnesium, and the caged calcium NPEGTA. What we seek to achieve is a step increase of calcium in that region, right by the ranitidine receptors, from 100 nanomolar, which is normal cytosolic calcium, to 10 micromolar. This is the level of calcium that we think develops during the brief opening of an L-type calcium channel. And so this simulates what an L-type calcium channel does to the ranitidine receptors in the cluster. We're just looking at one ranitidine receptor. That's the laser going off. It actually made a noise, not that loud. So anyway, when you do that, you end up seeing a step increase in the open probability of the ranitidine receptor. And this is just 3 samples, and what you'll notice is, and it seems pretty obvious to me, but maybe not to you, that there's a slightly higher probability at the beginning, slightly lower at the end. Maybe you see it best in the last record. You can do it with pKa phosphorylation using the catalytic subunit and do it again. And there's an even higher apparent open probability at the beginning and even lower open probability at the end. So when you plot these things out, and notice the time scale, it's pretty long where we look, you end up getting 2 records. No phosphorylation, rises to a peak and settles to a lower steady state level of open probability. And following pKa phosphorylation, it goes up nearly to 1 and falls to a level below the no phosphorylation. It's a complex, time-dependent series. We talked in some of the discussions about how the rate of change matters. And here's clearly a case where the rate of change of calcium in the subspace makes a difference to how the ranitidine receptor behaves. I would point out that the critical time for triggering an L-type calcium channel triggering a spark is that green band, something like a few milliseconds to 10 milliseconds in that time frame. That's roughly the time from the beginning of the L-type calcium channel opening to the closing of the ranitidine receptors during the spark. That's model dependent. I'll show you a little more there. There's another complexity that is worth pointing out. In the steady state, and again, all of this data is not entirely consistent. Okay, so I'm there's some problems there, but the data is what you end up having. If you compare the open probability curve black line under control conditions for ryanodine receptors with pKa phosphorylated ryanodine receptors, and you normalize them to try to see what happens at low and high calcium, you'll notice that there's an increase in open probability with pKa under resting conditions at low calcium, and sort of a decrease at higher calcium. So the complexity, this is for steady state, the complexities make the modeling exquisitely difficult. Nevertheless, there are a couple of rules that one can glean from that data. So the phosphorylation is complex and controversial. What about local depletion? This is also important both for modeling and the conceptual framework for how sparks work. So again, using flow 5N to load the SR with calcium, with a calcium indicator, measure the calcium. You can see this section of a cell is shown here. And again, like that image I showed earlier, you can see the T-tubules in cross-section, but now, not the T-tubules, sorry, the junctional SR in cross-section, now revealed as a dot. And you can see the line along the Z line for every Z line. And this represents the calcium load. How do you know it's a calcium load? If you do a line scan and then apply caffeine to cause a dump, a loss of calcium from the SR, you can see that the fluorescence goes away and it decreases. And this happens very quickly. This is done for the whole cell, but it certainly shows the details in the line scan image of a particular region that was scanned. The calcium blink now is a depletion signal associated with a spark. So I have to, and this is one reason I took the data, I told you I took some data out. We have some small sparks and small blinks, and these are somewhere between half and a tenth the size of the things I'm going to show you. You will appreciate how small the things I'm showing you are. So the reason I took it out is it takes a long time to present it, and that's the only reason. But you can see this is a blink on that same line scan image. It's kind of small, isn't it? It's not. You can see it pretty well compared to the things, the subgroups. But the blink is very clear, and it is made clearer if you subtract the background or if you do a computerized detection system looking for, differences that are significant in a region. You can signal average them, and you get a clear diminution of the signal. And you can follow this from the signal average signal where the nadir, the lowest point, can be measured. And this is about 30 milliseconds into the spark, or into the release event. And the half time of decay is about 35 milliseconds. That's the refilling function. So in some of the discussions today, we talked about loss of calcium during a spark and the refilling. And so this is primary data. Now, the data I'm going to show you is from one paper. We have other results. They're generally consistent. Some of the timing issues change from species and temperature and other conditions. So nothing is ironclad, but it's a question. Yeah.
Because
I need to drink something anyway.
I may be just more ignorant than everybody else, but just to make sure I'm on top. So you've shown us, you've looked at sparks. We all know what sparks are. Those are the release that you see into the cytoplasm from the SR.
This is the loss of calcium.
You're now looking at the SR itself, and what we're seeing is the loss of it. So the blinks, so you've got a lot of conditions and things where spark frequency changes, a lot of different genetic models and things. Do you see that the blink frequency matches spark frequency?
And so what I'm going to show you next is simultaneous measurements of sparks and blinks. Okay. So I was just, first of all, talking about this timing. So it's about 35 milliseconds. And the reason I'm mentioning the timing before I present the simultaneous records is this is roughly the kind of timing that you need for refilling models from the network SR to refill the junctional SR following a spark. A spark is a singular release event, and you have to account for all the fluxes of calcium. But in a global release event, timing is a little different. So and this is a single release event.
Is there information on the spatial dimension of the blink versus the spark?
Yes, there is. Again, something I cut out just for timing purposes. We've done some analysis of the spatial dimension. The blink, of course, is constrained spatially. First of all, its source is much smaller. The source of the blink is that pancake. And remember, the pancake should be almost completely found in the plane of focus. So it's about 25, 30 nanometers thick. It's 100 to 300 nanometers in diameter, and then folded around the T-tubule, which is 300 nanometers in diameter. So, depending on the details, that's sort of what it looks like. The spark, however, is a product of efflux and diffusion, facilitated diffusion of the calcium on the indicator around the site. So the full width half max of a spark, so that is the size of the spark, if you take the peak level and measure the width at half of the peak level, is something like 2 microns. And a point, again, discussion point that's worth keeping in mind for those who weren't party to it, is in all of the models that we've done, and as far as I know, others have done, the model spark is always smaller than the physiologic spark we measure. We haven't figured that out. So that's a good point of discussion. Don't understand that. Did I answer that enough? Okay. More than you wanted. So the junctional SR emptying continues after the peak of the calcium spark. In order to come to this conclusion, we measured both at the same time. We used ROAD2 to measure the spark in the cell, and Flow5N to measure the blink. You can appreciate, I'll just use this, I hope it doesn't blind you, but you can see that the blink is even harder to see when we measure both at once. Not surprising, we have to split the signal with a mirror, even though we signal average. It looks clearer on my computer screen, of course, but you can see it pretty well there. So you can see the sparks, and the size of the spark is pretty clear. And the blink on the analyzed data is really small, smaller than it was on the earlier trace, and that's just because of the loss.
I forgot, what is the difference in affinity of those two guys?
Well, ROAD2 is sort of like Flow3. Again, these are significantly affected by the viscosity and other environmental issues. So nominally it's like 400 nanomolar, 500 nanomolar. If you go through some calculations with protein concentrations, maybe it's 700 or micromolar, something like that for ROAD2. Flow5N is even harder to say. We've tried to calibrate that. That was something like 100, ballpark 100 micromolar. But we think, and so this is based on extension and a number of tests with different protein concentrations that we think are in the SR. We think the affinity shifts even more for Flow5N. We think it probably goes up to something like half a millimolar, as high as that. We think the concentration, again, this is, there are so many guesses here that it makes it very hard, but we think the concentration of calcium in the steady state in the SR is about 1 millimolar. But when you go through lots of calibrations without these corrections, you come up with a couple hundred micromolar, because you don't know what the affinities of the indicators are, just roughly. If you signal average the signals, you can see the spark and the blink, and you can see that the peak of the spark in this particular case, and this is what I was warning you about variation, the peak of the spark occurs before the nadir of the blink. Now, why do I even mention this? I mention it because there is, and this may exist here too, a naive assumption that somehow the efflux function somehow is related to the peak of the spark. Well, it is somehow, but not directly, because not just non-linearly, but you can imagine that you have a point source at the beginning that spreads through diffusion, and so you end up getting a rounding and a spreading of the function. And so the peak of the efflux, and we don't have efflux here, but I'll show you in the model, the peak of the efflux should occur almost at the beginning. And the peak of the spark occurs later as the flux diffuses. And in fact, most of the efflux functions are decreasing functions of time. And so that heightens that difference even more. And so pointing out that the peak of the spark is before the nadir of the blink just suggests that even after the peak of the spark is over, there is continued release, something that we think occurs, but this doesn't prove it because of the noise in the measurements. So calcium depletion in the junctional SR regulates calcium spark termination. So this is a model that we presented a while ago, and I'm bringing it up here in very short form just to try to tie all of that other data that I've just presented with the spark. The idea, with the depletion discussion. So the clusters of ryanodine receptors interact. This is the core of the model, and they gate cooperatively in what we call coupled gating. Coupled gating means cooperative. That's a probability coupling, not an absolute all or none coupling, as Andy Marks first suggested. The calcium content of the SR affects the open probability of the ryanodine receptor. So increases in the SR calcium content increases the open probability. That's sort of the underlying assumption, something that is now shared, I think, by many people. So in a simulated spark, this is our release function, and I would point out the time of the peak. So you can see what the codes are on the right: calcium release flux, SR calcium content, and the fluorescence signal, the calcium spark. Notice the peak occurs immediately with the opening of the ryanodine receptors. This is because when one ryanodine receptor opens, they all open all at once, according to this model. Some of these details probably are faulty. They probably should open a little more slowly than we have them open. The calcium in the SR depletes rapidly and nearly completely. And in our model, it's 90% depletion. Interestingly, even with 90% depletion and very low continuing efflux, if that had continued on for a long time, the level of the fluorescence of the calcium signal would be maintained at roughly a 50% peak level. And we had experimental data, again, that I don't have time to show that this in fact occurs. That you can get a long spark with a plateau level that's 50% of the peak fluorescence signal, and you can use various toxins, scorpion toxins, to demonstrate this, or ryanodine itself at low concentrations. So, so this is the release flux, this is the SR calcium content, and the calcium spark, and then the model spark with the image. Time to peak of the spark is clearly after the peak of the release, and the continued release occurs. And this shoulder is pretty clear on the model, but you don't see it in spark signals, and that's because there's a lot of noise, and there's a blurring due to the noise. So we'll leave the noise out now. Now I'll just point out a question.
You have a step function on the ryanodine receptor of probability, right? So that's what's giving you the shoulder.
No, the shoulder here is due to the fact that release is terminated.
Yeah, isn't it a step function on release? The open probability?
Well, actually, if you look at the details, you don't need a step function if you just get sputtering down. In fact, the details here, it's not shown too well on this plot. You can see noise as it falls, and there's usually a little noise, but it does slam shut. I mean, it's not quite a step function, but it's close.
What
to
is
it.
your driver function in the model? That's a step. Is that a step function? No.
The driver function, you mean
Yeah,
that?
above the calcium release flux, what's the driver function on the ryanodine receptor open probability that's driving that?
I mean, why does it open?
No, I mean, what
You want to
is-
know how the flux is driven?
No, I mean, above calcium release flux, that's in response to opening of the ryanodine receptor.
Right, so the way this works is one ryanodine receptor opens, and then they all are opened, and then you get full flux into the subspace that diffuses out.
So what you've just shown us with your hands, I guess you could have a white line that shows us what the PO of the ryanodine receptor is doing, and I'm guessing it's a step function. No, maybe not.
No, the PO of the ryanodine receptor, I don't have it plotted. We have it in the paper. What you see is that, in fact, it goes up very quickly. It sort of looks like a step function. This is what we've corrected in the more recent models. It's more of a stepwise increase. And then it sort of produces a little noise in the falling phase, a little bit as it flickers, and then a lot of noise later on. But it still has this overall shape. And the shape is driven by this combination of cooperativity among the ryanodine receptors and the subspace calcium, along with the depletion signal from SR calcium. So it's not quite a step function, but yeah, it looks something like that on the opening.
I just wanted to understand something about your comparison between the model and the experiment. The model you compute calcium concentrations.
Yeah.
And the experiments basically measure fluorescence.
Correct, right.
And if you try to analyze this, you mentioned before the disagreement between the spatial extent of the spark and
Yeah.
what you measure and what you simulate. If you try to look at the effects of the dye.
Yes. Yeah,
In
we've
a
done that, and
model- in a model?
that doesn't solve any of our problems. So we and others have done the same thing. So the first attempt at doing this with Greg Smith was really an attempt to do all of these investigations, and we couldn't get it to widen by any means. We played all sorts of games with dye affinities, dye binding, dye diffusion, asymmetric diffusion. We couldn't do anything that anyone could accept reasonably. And more recently, we've explored those again, same result. Leighton Itsu was able to get a wider spark if he increased the efflux enormously, so that he would actually saturate the signal in a small region, and then it would appear to be bigger, but of course it wasn't really. And there was more flux than could be accommodated or found in the junctional SR. So there are solutions, but I mean, if you have a good idea, there's probably a really good idea out there. I don't know what it is.
Yeah, but you rolled out the effects of
Yes.
the dye.
Yeah, in fact, the facilitated diffusion, we did a whole little section on that in the Smith paper. And it just, I mean, it plays a big role in the total movement of the signal, and the shape depends greatly on this flux and how the dye diffuses. The point that I was making here is that the number of ryanodine receptors doesn't significantly affect the actual appearance of the spark. So from 100 to 20 to 10, the spark is less. The efflux is proportional, the peak efflux, but the spark is largely the same over this tenfold change. So it is amazing, and this is due to the self-correction and the total flux. And you can see that with 100 ryanodine receptors, the point at which turnoff occurs is at a lower SR calcium content compared to, say, 10. So it's a self-correcting system due to the calcium sensitivity of the ryanodine receptor, that is, the SR calcium sensitivity. And if you plot the luminal calcium and spark rate, it forms a highly nonlinear function of luminal calcium. So SR calcium and function. So if there's a connection between the calcium content and the spark, then the reloading has to influence the spark, and so restitution experiments are helpful. You know, I'll do this quickly, but I don't mind any interruptions. When time is up, when you get bored, we can stop. So normally, if you just look at sparks as they occur spontaneously in a tissue, you see sparks that occur in different spots over time, about 100 per cell per second at rest in rat. But if you apply ryanodine so it binds to one of the subunits, you increase the open probability of that subunit a lot. And so it tends to open repeatedly in the same spot. And by doing that, you can look at how delay affects the height of the second spark. And so you can do a restitution experiment. And if you just do this experimentally, you can see the variation, small sparks and, for the second spark and fairly big ones. And if you do a full set, you can look at the restitution curve and how long does it take to refill and how does that compare to our other estimates. And, you know, a half time of restitution is something like 75 milliseconds. So it's still pretty fast, not 35 milliseconds, which we thought was the half refilling time of the junctional SR from the blink, but pretty close to that. So as SR calcium increases from a very low level, the sparks get restored and normally see coupling returns. At a high level, increasing SR calcium sensitizes the ryanodine receptors, and so it produces instability. And so that's what I had mentioned before. This was the same picture, same movie you saw before. And this is just due to the increase, we think, to the increase of sensitivity of the ryanodine receptor due to higher SR calcium content. So, calcium sparks and calcium signaling. So at higher SR calcium, the open probability is greater and the calcium sparks occur at a higher rate. pKa phosphorylation of the ryanodine receptor does increase the open probability, and as I said, the process is complex. And the complexity, about 10 slides that I cut out, really interesting stuff, sorry, about how you can actually see, you can unmask an invisible leak in the SR when you when you pKa phosphorylate the ryanodine receptors. And you can produce a whole host of other things, so that it's even more complex. But the important thing here is that mutations in the ryanodine receptor do predispose to arrhythmias with a higher opening rate when the open probability and all other conditions appear to be the same. So that's why, as I said in the introduction, we suggest that we need to talk about intrinsic behavior, P star, the intrinsic probability of the ryanodine receptor. And so, what are things that we have to deal with? So mutations could affect P star. We could say phosphorylation. Remember, this is a modeling notion as well as a conceptual notion, that you can change the intrinsic opening probability of the ryanodine receptor by some means.
Can I give you a term? The intrinsic open state stability instead of P open. P open is operational at any point in time,
Right.
but you can have the intrinsic open state stability, which will determine the open probability given the input parameters. Just another phrase, another term.
It's an idea. It's an idea. I better think about it.
Yeah.
Okay.
I think that's what you're talking about,
Well,
right? Essentially.
we can do this over dinner. I'm not sure why it would be helpful to add stability as I agree
open
that
state stability, because it's not actually the open probability you're talking about. You're talking about the intrinsic ability to have an open.
That's right. That's correct. Okay.
We'll talk.
I agree. Calsequestrant mutations produce changes in P star. Blockers of the ryanodine receptor, like tetracaine, widely used now, change P star. Sensitizers like caffeine change P star. So P star of the wild type ryanodine receptor, just as an example, would be less than P star of a catecholinergic polymorphic ventricular tachycardia mutation. It's a mutation of the ryanodine receptor. So P star. So let me talk you through some of the terms. So P star is the open probability is, I'm not sure what term The open probability could be equivalent to the probability of getting a wave or the probability of seeing this massive calcium signal, which we've called the tsunami, or spark. They're all sort of interrelated and proportional. The SR calcium content concentration is on this axis, and on the other axis is P star. Now, again, some people find this intuitively obvious, others don't. So stop me if it's a problem. So this is the basic plot that we imagine. P star increases at any particular SR calcium content, you get an increase in the open probability. That's the vertical axis. At higher SR calcium contents, that increase is steeper. Okay. So those are the equivalent terms. So this plane that is drawn here in red shows one SR calcium content and how the open probability changes as a function of P star. Perpendicular plane shows at a constant P star what SR calcium content does. So as SR calcium content goes up, the open probability goes up. So if we take a given P star, given intrinsic sensitivity of the ryanodine receptor, and plot the curve that it would follow as a function of increasing SR calcium content, let's take a spot when the SR calcium content is completely empty and see how it changes. So as circa pumps calcium in to that empty SR, the open probability goes up as the SR calcium content gets sensitized. Now it doesn't have to just be circa. It could be diffusion in from the network SR. So whatever it is that leads to the increase. When a spark occurs, or when the ryanodine receptors open to release calcium, then you expect this to rapidly fall and go back to the state it was at before. Now what about circa? So it pumps calcium into the SR. Any questions? If circa behavior is unchanged, then the rate of loss of calcium from the SR, the rate of loss through permeation pathways like ryanodine receptors, the rate of loss determines how much calcium there is in the SR. So this plane is what we would call a circa plane. So it's a plane that's established by a particular state of circa. And hopefully you'll agree with me that that's the case. So let's take a couple of examples. So we'll take what tetracaine does. We start off with a point in the circa plane that is in steady state balance. So the pump and the leak globally are in balance. So I would point out that all of the SR and all of the ER are interconnected. So if there is leak one place, it equilibrates in the steady state everywhere. And then when you apply, the instant you apply tetracaine, you decrease P star. And so the open probability suddenly falls. But at that lower P star, circa still works, the leak is down, tetracaine was applied globally, and the net effect is that over time, circa pumps calcium back into the SR so that pump leak balance is reestablished. And it's going to be reestablished at the same plane because that gives you a leak that equals the circa influx pump. So what David Eisner, Hector Valdivia, and Don Burr said is right. In the steady state, if you change the sensitivity of the ryanodine receptor, it's self-correcting in steady state. What happens when caffeine is applied? And these experiments were published by Trafford and Meisner recently. So it's the same sort of thing. You start off in the circa plane. Instantly, when you apply caffeine, the open probability goes way up because P star is higher due to the caffeine. When the release has occurred, as it will, then the SR empties, and in the steady state, Circa pumps it back in until there's a pump leak balance. So the pump, Circa, which is unchanged by all of these, is constant. Now the leak compensates for what the pump does until the SR calcium is at a level that permits the pump and the leak to be in balance. So what happens if you have a higher level of Circa? You get a higher plane. So Circa is pumping more quickly. And so if you start with a normal Circa and suddenly increase Circa or do it, then you get a transition from one position to the other. So what about if you compare steady state conditions at a normal P star, and so you have a certain SR calcium content at this P star, and you're in one Circa plane, and now you have a higher P star, so you have a different SR calcium content, lower SR calcium content, but you're in the same Circa plane. And now if you end up comparing those two, the difference isn't in the open probability. The difference is in the SR calcium content. If you now do something that will load to the same level, the SR, then what happens is you can manifest different open probabilities. And this is what we think happens in the case of these arrhythmias. You can transiently increase SR calcium. And that effect of transiently increasing the SR calcium is different at different P stars. And so you have a high probability of release when P star is high, and a modest increase when P star is low. And so the consequence of different P stars isn't in the steady state, it's during the transient elevations. And when would this be manifest most? Probably not with steady beating, because with every beat you unload the SR and then it's restored. It would be in the pauses between beats where this higher open probability would have a chance to discharge and trigger neighboring release sites through propagation of waves. So it's the same what I just said. So the factors that need to be considered in the modeling is calcium influx, NCX efflux, plasmalomal calcium ATPase, Circa forward rate, Circa backward rate, a lot of things that we all model. SR invisible leak, the thing I had to take out, but it's real and substantial, particularly following pKa phosphorylation. Other buffers, and for example, the mitochondria, depending whose model you use, mitochondria may play a really important role in buffering calcium. This is another controversial area, not to be discussed right now. And of course, P star is important in arrhythmogenesis. So I thought I'd end with 2 or 3 comments. The P star, with additional stimulation, if we change the heart rate on beat 5 with a high P star, you can actually, with lower rates, get more leak than you get load. And with higher rates, you can increase the SR calcium load and hence increase the probability of getting a wave or an arrhythmia. And when you go to very high rates, you may first decrease and then increase just because you stimulate release with each beat, and it takes a while to build up the content. So you can get complex behavior at different rates, and if there's a pause when the open probability, the probability of a wave is high, that's when it probably is most likely to be manifest. So the luminal calcium is important in regulation. Cytosolic calcium and SR calcium can change in the steady state and transiently, dynamically. In addition to SR, there are lots of factors that regulate open probability. And when trying to resolve this issue of how calcium overload arrhythmias may produce SR calcium produce arrhythmias when SR calcium is normal or low, I think the concept of P-star or some intrinsic Brightening receptor opening probability is quite useful. I think this is helpful not just with ventricular myocytes, but also with atrial myocytes, and we have some data on that. I do have a final thought that may be of interest to some. You know about the Vasa? Anyone ever hear about this? Was a warship. So the Vasa was a Swedish warship built in 1626, started to be built. And it actually, in its maiden voyage, sailed about 1 mile and sank. It's a classic story in Sweden. And this is a picture of the Vasa. And if you look at these ships, you've probably seen these things on your TV and other programs. They always look unstable. They're too tall for their width. Well, this was redesigned top down by the king. Made it even taller. So, what happened was, there were unforeseen leak pathways. These were the gun ports. And when the thing heeled over in a light wind, it heeled over enough to fill with water through the gun ports. Thank you.
Any questions? I think the P-star, it's basically just the intrinsic ability of the Brightening receptor to open.
Correct.
And that's what it is. So with phosphorylation applications, it has a greater intrinsic ability to open. It's not the probability, because it's not.
That's
a
right. It's
probabilistic
intrinsic.
term. It's intrinsic
I agree.
ability.
I agree
Ability
completely.
of
The only reason I
on
made
the Vasa thing, there is a museum in Stockholm. Those that are interested, there is a replica of this
Right.
boat.
Yes.
Real
Well,
size.
no, it's not a replica.
It's the
It's
real
the
boat.
real boat.
It's a real boat.
Yeah, well, I was going to show
It's the real
pictures of
boat.
that and the gun ports.
You can walk through the boat. It's really interesting.
The thing flipped over. I mean, it's hard to believe in 1 mile. But the
I
reason.
thought all of Sweden was watching.
That's right. The reason I mentioned the P-star, though, is I think it isn't used as a free parameter.
I think it's less confusing if you call it just the intrinsic capability of the receptor
Well,
to open, and not just the probability, because the probability depends on all the other conditions.
Well, that's right. Well, I called it P-star and called it intrinsic probability, but you know, intrinsic
capability
opening.
or intrinsic ability of
Yeah.
the
Well, I agree.
Not probability, because it's not.
Okay.
I think now I understand the basis of Sweden's subsequent world domination as a naval power.
Well, apparently they were fighting Poland at the time. So you take it from there. Okay, well.
So it's surprising to me that those dyes don't affect the distribution of the spark.
Well, the dyes certainly have an effect on how calcium gets distributed, because if you look at, you can calculate what the calcium distribution would be without the dyes, and it's different. Not very different, but a little different, because there's facilitated diffusion of the dye.
So then the question is whether it affects the open probability of the receptor, because
It probably
there
does.
are buffers in the restricted space also.
Well, since it affects diffusion of calcium,
Yeah,
then
yeah.
it does. Yeah.
So I'm not really sure if I understand the concern with things at steady state, like this negative feedback.
Yeah.
So it would cancel itself out, but don't arrhythmias happen with a trigger, like it happens in a transient sense. So you explained it in that way, but the argument, like, I think I'm missing something.
Well, the reason I sort of beat it to death was the fact that there have been something like 30 papers in the last 2 years that have argued back and forth whether the increased P-star, the intrinsic opening rate of the Brightening receptor, can be responsible in some way for sudden cardiac death in catecholaminergic polymorphic ventricular tachycardia, a real cause of death. And the argument has been, if you do steady state experiments done by 3 or 4 notable scientists like David Eisner, but others, I mean, good colleague, if that accounts for it, and what he finds is that you apply caffeine, you increase the open probability, you get more sparks, and then it comes back to the same spark level, and he was using this to conclude that the answer is no. And you can say that's silly, but the reason I pointed out is, you know, as a modeler yourself, as someone thinking about this, you have to take this into account, because this is going to be the group of people who are going to be reading and reviewing your papers, and also writing long reviews or your grants.
But this is almost self-obvious, because
It seems obvious to me.
Because then, you know, if steady state, if the steady state situation was the cause of arrhythmias in general, then we'd probably all of us be dead by now.
Of course. Well, dead or yes.
The key
No
is
blood
that
flow.
the key is that it doesn't happen on steady state, but it happens on all of those dynamic
Correct.
transitions, and then
Yes.
all of these conditions have to come at, that's the obvious question. Why does somebody has lived for 35 years and all of a sudden on Thursday at 8 o'clock he dies from a sudden death and an arrhythmia? It's because all of those conditions have to come together on a very dynamic transition.
And it also suggests that the probability of the confluence of these conditions is very low.
Yeah, of
Because
course.
frequently people are 50 years old before they die.
Yeah,
So,
high probability genes,
right.
people died already.
So, again, it sort of calls to Newcastle in a way here, but not everyone, I think, in the audience is probably aware of the way this debate has evolved. And so, if that is what I'm bringing to you, that's useful too. To be aware of it, but I think for me it was entirely obvious, and I thought it was unnecessary to even discuss it until I too was caught in several debates. You know, just so it can happen to you too. Okay.
We're lucky that our steady state is pretty stable and free of arrhythmias.
Well, but also, you see, they don't always point out that it's steady state the way I did. So, anyway,
Any other questions or comments?
bring to the audience. Thank you.
Thank you very much.